Okay, so again, we can kind of set ourselves up. Draw a big cell.
Do you want this one dotted or no?
There's no dotted lines. The nuclear membrane is now gone.
I'm not gonna let these ones fall, I promise.
No. Okay. But I am gonna draw my centrosomes and my centrioles. Okay.
Okay, I'm
Okay.
Little circles.
Ready, Andy? Yeah, circle, squares, and yeah.
Mine.
Okay. Are we drawing this? We got this. All right. Now we are going to draw something dotted in this one, but not a circle. Okay.
Octagon.
What
triangle.
word did we learn yesterday? What vocab term do you think would fit nicely in this quadrant?
Trapezoid.
Circle.
No, I'm not looking for a shape. I'm looking for a vocabulary term.
Metaphase plate.
Absolutely, Philip. So down the middle of the cell, I'm going to draw a dotted line, and this is going to represent the metaphase plate. Now, is this plate something that I could actually touch?
No.
Is it a solid structure?
Nope.
No. What is it? How can we describe it?
It's an
Liquid or
imaginary
gas.
structure.
Because you can't get solid.
Okay, I like that line of thinking, but it doesn't actually exist.
Oh, so
Okay,
it's nothing.
it's an area. So yesterday, what did we talk about? It kind of very similar to being
An equator.
an equator, right? The equator on our Earth. Okay, bless you. Shanice. It's an area rather than an actual thing. Okay, it's not something we can touch. Same idea. This is the center of the cell, but not a structure that actually forms. Okay?
What is this in part of your body? What part is it in your
Every
body?
single
So like if you just like cut your hand off and
you
I'm
would be
guessing
losing
I don't
a
get
blood
that. Yeah,
is that a
I
blood
know
cell? Why
What
can't you just
cell
put
are
your
we
blood and
talking about?
basically on
I'm
your
ruin my
that?
back.
That's so pretty.
essentially.
Yeah.
It's a generic
Okay.
cell.
Okay.
Yeah.
When you see a droplet of blood, there are hundreds of blood cells
Stephen
in
Yeah.
that droplet. Okay. So, guys, I understand how this can be a little confusing because it's so small.
Yeah, the million.
Okay. We have trillions of cells in our bodies, and the vast majority of them are going through this all the time.
Like right now?
Okay, like right now. You have
Like
skin
me
cells
moving my arm
going
like
through
that.
this right now. Like we could swab the inside of your cheek, get some cells, and
Really?
see
That's
some
what
in
the
different
SVU
phases
people
of
do.
the cells.
That's
Let's
what
see.
they
Okay.
Yeah, they
While
do.
they melt.
Okay.
Right here real quick.
Let's stay focused, all right? So
Okay.
now,
Swallow
you can
melt.
do whatever order colors you want, but this is important. They must stay in that order for this to make sense. Okay, so I'm going to kind of put them in the order I have here. I'm going to draw my pink one on top. Notice,
Yes.
half of the chromatid is on one side,
Yes.
half is on the other side. Okay,
I'm just gonna draw X's, is that alright?
whatever floats your boat. Stay consistent, okay? And then it kind of looks like green was next. Notice that not all my chromosomes are the same size, and in real life, that's true. Okay, not all of our chromosomes are exactly equal.
Is this okay?
Okay. Now, I have to draw in something very important. Okay. And what did I draw in my first phase that I have not? Well, I drew my centrioles, right? They're hanging out over there.
But they're circles.
You're right. They should have been squares to be consistent, but
The hairs.
the hairs. What are the hairs?
Spindle.
Spindle fibers. Okay. What is the center of every chromatid?
It is the base. No, it's the I got the centromere.
It's a tree.
It's
Awesome.
button centro.
You're right. It is a centromere. A spindle fiber attaches to each centromere. In your pictures, it should be physically touching that center. Perfect.
I knew it.
Good. Good.
I'm a genius.
Okay. It attaches right to that centromere. Okay.
Making that money.
So if we wanted to describe what was happening in this phase, what would you say? What is different between this phase and that one?
They're ripping. They're forcing. They're splitting.
Are they splitting for you?
No.
Well, I say this because the circle's gone.
What is the only thing that has happened? Okay, so the nuclear membrane is fully gone. Absolutely correct. Where did my sister chromatids migrate to?
To the middle.
The middle.
To the middle. Okay, so my sister chromatids line up at the metaphase plate.
So are they replicating?
You answer that question for me. Are
No.
they replicating? Are they replicated already?
Yeah.
Yeah.
Yes.
Okay.
Okay? This is a great question. If my DNA was not replicated, this is what this would look like.
Uh-oh. Half.
It would only be one side of that chromatid, right? During the S phase, we make
The Dairy Queen.
the other side. Okay, we make that duplicated chromosome. All right? So during metaphase, all they do is line up. Okay? Now I'm going to draw an arrow going down. Okay, so we're going to stick with this